You know, our DNA is basically the code of life. It's the software that runs every single cell in our bodies. For decades, scientists have dreamed of being able to go in and edit that code to fix the little bugs that cause so many diseases. Well, today, we're going to dive into the technology that's making that dream a reality.
Just think about it for a second. So many genetic diseases, from sickle cell anemia to even some cancers, are basically just typos in our biological code. A single misplaced letter out of billions can have absolutely devastating consequences. So what if we had a tool that could find that one specific typo and just correct it?
Well, that's exactly where CRISPR comes in. The easiest way to think about it is like a find and replace function, but for our genes. It's a system that has completely revolutionized genetic engineering because it's so precise and, honestly, pretty simple. It gives us this incredible ability to rewrite the very instructions of life itself. So let's see how this thing actually works.
So, how does this biological word processor actually do its job? Well, at its heart, it's a surprisingly simple system with just two main parts.
Okay, the version you hear about most is the CRISPR Cas9 system. Now, don't let the name scare you. Its power really comes from the simplicity of its two key components. You've got a cutting enzyme and a guide molecule. Let's break down what each one does.
All right, first up is the Cas9 nuclease. This is really the workhorse of the whole system. The absolute best way to think of Cas9 is as a pair of molecular scissors. Its one and only job is to cut right through both strands of the DNA helix, making a nice, clean break exactly where it's told.
But how do the scissors know where to cut? I mean, the genome is huge. That is the job of the second component, the guide RNA, or GRNA for short. The GRNA acts like a genetic GPS. It's this small piece of RNA that scientists can program to match any DNA sequence they want to target. It finds that one precise spot in the genome and tells the Cas9 scissors, okay, cut right here.
So we have our scissors and we have our GPS. Let's see how these two come together to actually perform the edit. This is where the magic of CRISPR's precision really shines.
It's really just a simple three-step process. First, the guide RNA, our genetic GPS, scans through the massive genome and finds the exact DNA sequence it's been programmed to match. Then, once it finds it, the Cas9 protein locks on. And finally, the Cas9 scissors do their thing, cutting both strands of the DNA. This creates what's called a double-strand break. And this break is the crucial first step to making any kind of edit.
Now here's the really cool part. Making the cut is only half the battle. The CRISPR system doesn't actually write the new code itself. Nope. Instead, it takes advantage of something incredibly powerful, the cell's own natural DNA repair machinery. As soon as the cell detects that double-strand break, it basically freaks out and rushes to fix the damage.
And it turns out, the cell has two main ways it can fix this kind of break. The first one is kind of like a quick and dirty patch-up job. It's super fast, but it often leaves a few little errors behind. The second method, though, is much more precise. If there's a correct DNA template nearby, the cell can use it to make a perfect, flawless repair. And scientists can cleverly use both of these natural repair systems to get different results.
So by triggering that quick and dirty repair, scientists can effectively knock out a harmful gene. Those small errors that happen during the repair are often just enough to disable the gene for good. It's kind of like snipping a faulty wire to shut down a malfunctioning circuit. This is incredibly useful for turning off genes that can drive diseases, like certain cancers.
But what if you don't want to break the gene? What if you want to fix it? Well, by using this cell's precise repair pathway and giving it a correct DNA template, we can guide it to fix a faulty gene. The cell literally uses our template to replace the bad sequence with the right one. It's effectively correcting a typo in the genetic code, which could treat diseases caused by just a single mutation.
And we can go even further than just fixing typos. Using that same precise pathway, we can not only correct a gene, but we can also insert a brand new piece of DNA. If we provide a template with a new genetic sequence, the cell's repair machinery will just seamlessly integrate it right at the cut site. This is like pasting a whole new functional sentence right into our genetic document.
Okay, so we've seen how CRISPR works on the tiny molecular level, but let's zoom out. What does this all mean for us, for human health? Well, the impact is nothing short of revolutionary. We're really talking about a whole new era of medicine.
This quote here really just captures the weight of what we're talking about.
For the first time ever, a scientific dream that people have had for ages is actually becoming a clinical reality. We're moving away from just treating the symptoms of genetic diseases and toward fixing them at their very source.
I mean, the potential applications are just breathtaking. We're talking about the real possibility of a single-shot curative option for disorders that we used to think were completely untreatable. This technology lets us permanently correct the mutations that cause inherited diseases. It lets us fight cancer by disrupting the genes that fuel it, or even restoring the genes that are supposed to suppress it. And it's paving the way for a future of truly targeted and personalized medicine, tailored to your own unique genetic profile.
So the CRISPR revolution is here, and it has given us a tool of immense power. The ability to edit the fundamental code of life holds this incredible promise to cure the incurable and to literally reshape our future. But of course, it also raises some really profound questions about where we go from here. With the power to rewrite our own biology, the question is no longer if we can, but how we will. What story will we choose to tell?